We are now going to focus in on oxidation of the fats themselves. And so we've eaten lots of things, we've seen how we make ATP, but we haven't actually extracted two of the three energy sources, and quite frankly, the most intense energy sources, although both of them are extracted much more slowly. I realized as I was building the slides for this one that we actually repeat a lot of things that we just covered. So I deleted like 4 or 5, maybe even 6 slides in the beginning of this, because it was just a repeat of what we just looked at. And when I was prepping the notes, I prepped the chapters in a different order, and so I kind of repeated things. So what we do in terms of this, the types of fats that we have to deal with are the ingested fats and with the fats that are mobilized by the adipose tissue itself. The fats are carried around in carriers, a membrane that has the fats inside of them that are carried around, or they're carried in albumins to the site where they're going to be used. The gallbladder, this is why I left this particular slide on. The gallbladder is there to help you digest fats. And this is where the cholesterol collects, and this is where the bile salts are introduced to make sure that the fats that you do eat are solubilized. In terms of gallstones, they are collections of cholesterol. And so if someone develops gallstones, you have lots and lots of cholesterol. I know that Dr. Brown in organic chemistry, one of the experiments was to go down to the hospital, get gallstones, and then bring them back here, and you guys extracted cholesterol for many years from gallstones and how much cholesterol we could actually get. I don't know if they do that anymore to extract the cholesterol that way. Is a gallbladder necessary? No. Have you heard of people having gallbladder surgery? Let me tell you, I had some gallstones at one point in my life. I thought I was having a heart attack when I started feeling the pain. It was not fun at all. I was here teaching in the middle of the day, and I was going to the hospital in the middle of the day, calling my wife saying, I don't know what's happening, but I'm feeling poor. So I did all the heart stuff. That was fine. We found out it was the gallbladder. And you know when you go to tests, you see a technician first to actually do the test, and they'll never tell you anything because they're not allowed to tell you, even though they see the scans all the time. A doctor has to look at it and then interpret what they see. Well, after I was done scanning, I wanted to ask the tech, what did you see? And I just don't, because I know they can't tell me anything. But he looked over at me and he said, so how much pain are you in? And I thought, if he's asking me that, he must have saw something that didn't look quite right. And so the number of gallstones that I had and the size of them were really big. And I was just miserable. We got into surgery fairly quickly, and oh my goodness, it was like night and day. I went from miserable to feeling so much better, having the gallbladder taken out. Here is what most people suffer after the removal of the gallbladder. If you do eat a fatty meal, then it tends not to be able to be dissolved as well. And so the fats are supposed to be soluble and are supposed to be absorbed in your intestine, now get in the small intestine, is now an oily mix, and it goes through that way and not so good at the end, okay? And so people that do have gallbladder surgery have to adjust their eating pattern. They are not as tolerant to very fatty meals. So say goodbye to Taco Bell and fourth meal. I left this particular slide in. We did see this last time, that once you have the hormonal response, you get upregulation of the lipases in adipose tissue, and then the serum albumin will hold up to 10 molecules of fats that are free and take them to the muscle cells. And that's really where most of the fat is broken down, is in the muscle cell. And once free fats get into the muscle cell, really the only thing that you can do to them is take them down the metabolism that we're going to take a look at here. What we will eventually do is merge the fat into the citric acid cycle and then going to the oxidative phosphorylation. And so it's going to end up being the same molecule as we derive glucose. And so glucose eventually made acetyl-CoA to go into the citric acid cycle. We're going to make acetyl-CoA from the fats. Now, the energy that exists in fats is huge, okay? They're more reduced, they're more anhydrous. And even carbon to carbon, if you by chance take, let's say, an 18-carbon fat and you take 3 glucose molecules, you'll actually extract much, much more energy, more ATP from the fat than you will from glucose, even though we're talking 18 carbons to 18 carbons. And so they are very reduced, and every oxidation extracts electrons, which pumps out more protons. And so when you take a look at this, all of those carbons have to go to CO2, so we have to go through several oxidative steps to get there. In terms of the energy formation, with every fat molecule in the adipose tissue, you have 3 fats that are connected through ester bonds, and you have the glycerol that can also be oxidized through entrance into glycolysis. In a generalized person, in which I don't know anybody who is generalized, okay, the 70-kilogram person, if you take a look at where the energy stores are, glucose is what we really want to use. That's the fastest, and then glycogen. But look, that's 640 calories versus protein and fat, where the majority, I mean a vast majority of your energy resides in proteins and fat. Now, even though that numerical value of proteins is very high, the body tends not to use all of your protein, because that would also degrade the muscle. You won't be able to move around, and if you can't move around, then in a starvation condition, you can't go forage for food very well. And so it will take protein, but it tries to conserve that as much as possible until you get very, very low in energy stores. What happens in the lipases is adding water across the esters to get the fats, the free fats, and glycerol. The glycerol can be converted into intermediates of glycolysis using 2 enzymes, glycerol kinase, where the hydroxide attacks the ATP at the alpha, beta, or gamma phosphate. Gamma, because ADP is the leaving group and I transfer 1 phosphate. And then the hydroxide in the second position here reacts with NAD plus and a dehydrogenase, and so I will allow the conversion of the hydroxide into the ketone. NAD plus will be reduced, this will be oxidized, and then I'm in a glycotic intermediate. The fat degradation, though, took a little bit more to find. And I don't want to be the grad student for this professor, okay? Because this is what he did. He did find this. He found fats are degraded by 2 carbon units. Okay, that's what he did. But the experiments that led to this were feeding dogs phenylpropionate and phenylbutyrate. Okay? So inside their food, they gave them these organic molecules, of which they had short fats attached to benzene rings. Okay? And they wanted to see what happened and what was the outcome. So they had to wait to look and extract the outcome out of the waste product. So some graduate student had to collect the urine off of these dogs and then isolate what was there. Not a way to get a PhD that I want to do. Okay, this is the outcome of what they found. If you started with the phenyl butyrate, and so you have 3 carbons, benzoate is the outcome. You lost 2 carbons, and that was the oxidized species that was excreted. If you look at the 4-carbon intermediate, you lose 2 carbons, and you're left with the phenyl acetate as the outcome. Each time, 2 carbons were excreted or broken off, and then you get too close to the benzene ring where it is not a free methylene, and the benzene ring prevented the enzymes from continuing their degradation. So depending on, it didn't matter which fatty acid they used, every time 2 carbons were lost. Okay? And so whether it was odd or whether it was even 2 carbons were lost. So the terminology that he used was derived from the, how we number our carbons. In the formal numbering, the acid is considered the 1 carbon, and then we count out to the end. We also designate with Greek letters. This methylene is alpha to the acid. This methylene is beta to the acid. And then one other Greek letter that we use is omega. You may have heard of omega fats that are good for you, and fish oils have these. And so what the terminology we use in degradation of fats is what's called beta oxidation, because this carbon will begin as a methylene and end up as a ketone in the final product. Now, unfortunately, the fatty acid comes off as a free acid. It will react with the ATP, and it actually will attack at the alpha carbon. As you can see, we haven't seen a lot of that, but we have seen once or twice attacking at the alpha carbon. What we haven't seen very often is the beta attack. It does exist, but not a lot of enzymes do that. The artist forgot a couple of electron movements. Okay, and we have the fatty acid attached to AMP. And we give off, a lot of times they'll write PPI. The pyrophosphate is never found in the body because we have a generalized enzyme called pyrophosphatase, which will break them down into 2 individual PIs. That's an equivalent of a phosphate. And then we will have the CoA come in as a carrier group. You have to have a base to take off the proton, and then the attack. You have to kick up and you have to kick down. I'm just so disappointed sometimes when I look at mechanisms drawn out in biochemical text. And AMP is the leaving group. So essentially, we attach the fat molecule to a CoA, and we end up with AMP. This takes 2 equivalents of phosphate energy. And we add this fat onto CoA because CoA is the molecule that brings whatever it carries to degradative enzymes. And so when you put something on a CoA, most likely it's going to end up being a degradative process. Here's the overall degradation. A fatty acid plus an ATP will become an acyl CoA, AMP, 2 PIs, and 2 protons. For a long chain fatty acid that links to CoA, it's called acyl until you have 1 methyl, when it becomes acetyl CoA. And so anything past 1 methyl group becomes acyl. Okay, so once we have gotten that fat inside the cytosol, we bring it in and we have to add the CoA onto it, and the problem is degradation happens inside the matrix of the mitochondria. I've got this long fat attached to a CoA. I've just spent a lot of energy to add it on, and the first thing I have to do is take off the acetyl-CoA because it can't be transported across the membranes. So the acyl-CoA will react with a molecule called carnitine by acyltransferase version 1. Every time you see version 1, what does that mean automatically? There's at least a version 2, if not lots of other versions. Here, the carnitine will have a reactive hydroxide right here, where the acyl will attach. Simply a base will take the proton. I will then take the electrons, attack the carbonyl, kick up, kick down, CoA falls off, and I get acylcarnitine. Now, if you take a look at that, that molecule is a lot more charged and polar than what we just had. Okay? I'm trying to get through a membrane. The advantage is this. There's a transporter, a protein that will bind to that molecule. And it will bring in a acylcarnitine. In this case, they actually indicate that the fat was 16 carbons long in that particular acyl version. And it will bring it into the mitochondria. It is an anti-transporter such that it can only bring in the fat when a carnitine that has no fat is transported out. It keeps the balance of the carnitine on either side of the membrane. And once we get in, we have carnitine acyltransferase version 2 that is found only on the inside of the mitochondria, which then will put it back onto CoA. And so the 1 puts it on the carnitine, the 2 will make it go back to the CoA. And so I now can do my degradation. Took a little bit of energy to get that inside. 4 steps to get this done. 1, 2, 3, 4. All right, we're going to oxidize. We're going to hydrate. We're going to oxidize. We're going to thialysize. On that list, what word is new to you? Thialysis. What's lysis? To cut. What's thio? Oh, SH, sulfur. So a lot of times we've heard hydrolysis, where an OH reacts and breaks something. This is thialysis, where SH, which if you look, oxygen number 8 and sulfur number 16 are in the same group. They react very similarly. All right? So we will do this, and we will do this many times to degrade a fat. Okay, first enzyme is acyl-CoA dehydrogenase. Uses FAD. If you have a dehydrogenase and an electron acceptor, what leaving group will you have in the mechanism? Well, you'll make FADH2, but what's the leaving group that you can invoke? That if you wrote this leaving group in on an organic exam, you will be marked wrong. Leaving groups are things that like to leave, like phosphates and AMP and even hydroxides. But a hydride. Yeah, hydride's not really stabilized by anything. It's a really high energy leaving group. Tends not to happen in an organic flask. You really have to force it and have really extreme reaction conditions. But in the body, it happens all the time because I have electron acceptors. And so I'm going to use FAD, and we usually will convert a straight-chained hydrophobic portion of the molecule and create an unsaturation in that hydrophobic area. And so we go from fully saturated to a double bond. And we have 4 hydrogens at the beginning, and we only have 2 hydrogens attached to the product. So in organic mechanisms, what do you have all the time, every time to use? You always have water. What else? You can always invoke an acid, and you can always invoke a base inside an enzyme because they are side chains that are placed in the active site. In this case, do you think I'm going to use an acid, or you think I'm going to use a base in our first reaction? Base. Because in reactions that occur inside the body, it's all about the base. All right. I used to say that all the time, and then this song got in the way. All right. I have 2 opportunities. I can attack either the hydrogens at A or the hydrogens at B to deprotonate. So A or B. Knowing what you know now and reasoning through this, okay, the organic intermediate is going to try, and the enzyme will try to create an intermediate that has the lowest energy value so that it helps stabilize. So I want to try to use the advantage of the organic molecule and its structure to lower the transition state. Will I take a proton from the A position, or will it take a proton from the B position? Okay, think about that. Okay, you vote what? I got 1A. What do you think? A there. Say A. A because everybody else said it? Yes. Brittany, what do you think? You'll say B. Ah, we've got someone who wants to walk a different path. You think it's B? Oh, wow. 4-3. B, 4-4. You're the tiebreaker right now. A? A. Okay, we have A as the intermediate right this very moment. All right? Okay. The mechanism can be written very similarly. All right? You can either do this, and I will write the A and then I will write the B. Here's the A. I would kick down, and this would be the leaving group to FAD. I will write B in a different color. I will go here, I will go here, and I will go there. The difference between the two is the first arrow. Okay? In the first arrow of the blue one, the proton is deprotonated. The electrons sit on the alpha carbon for just a tiny bit of time. Okay? In the first on the green, the first arrow, the negative charge sits on the beta carbon. Okay? So I have a carbanion here and I have a carbanion here on the alpha or the beta. Okay? One is organically more stable. It is the alpha because you can form a resonance structure with this ketone. That is the rationale why you're going to attack at the A's, the alpha. Okay? If you're alpha to a carbonyl, you're a bit acidic. And I create the trans fat in the 2 position. All right? Essentially, I've already synthesized a bit of energy. All right, this enzyme, now that we've taught oxidative phosphorylation, can be also named complex 2. Okay, in oxidative phosphorylation, you had complex 1, complex 2, 3, and 4. That enzyme is complex 2. All right, we are now going to hydrate. It's always important to keep hydrated. All right, hydratase. I love that enzyme name. All right, and you obviously know that the hydroxide is going to attack at the beta because the hydroxide in the product is at the beta, so it must attack right there. So I will take and deprotonate the water. The enzyme will. I'll attack, and I'll pick up a proton. The intermediate on the third arrow, while I have a proton ready to take, if you had a transition state, the negative charge would be on an alpha carbon to a carbonyl, so it could be stabilized. All right, I have hydrated. First reduced, then I hydrate, and now I'm going to oxidize one more time. I will again use an enzyme called a dehydrogenase. I have a different electron acceptor, NAD+, and NAD+ is used quite more often to oxidize a hydroxide into a ketone, whereas the FAD is usually done on a fully saturated fat. Then I now have the two hydrogens. You have this particular position that's attached to the carbon and this particular hydrogen that's attached to the oxygen. Either one the base can attack. Would you think that the base will attack the one that is attached to the carbon or to the oxygen? Oxygen, I got one oxygen. Go ahead and tell me what you think. Blurt it out. Oxygen, two oxygens. Anybody else? Okay, I got two that are brave, and I got a whole bunch that are just going to guess, and they're going to be 50% right 50% of the time because it's a choice. If you, here's the mechanism. I will deprotonate the one on the oxygen because the electrons can be in a negative charge sitting on the oxygen who is very electronegative. The other attack would leave a carbanion. On the beta, there is no stabilization at all. And so this will kick down, and this hydride will be accepted by the NAD+. I have now converted energy. At this moment in time, how many ATPs have I synthesized, knowing the stoichiometry of what can happen when you produce electron carriers? Oh no, I'm incorporating the last exam into this one, or even the first one, first exam into this. How many ATPs have I synthesized effectively? Because remember, the organic molecule is potential energy, right? And we're changing it into other forms of energy. These were electron carriers, which then get converted into a proton gradient, which gets converted into ATP. How many ATPs have I synthesized? Okay. Now we have created the potential to convert them if we allow the electron carriers to go through the oxidative phosphorylation. And we've created them inside the mitochondria, so they will go through oxidative phosphorylation. For every FAD produced, how many ATPs can I make? Oh, you answered this on an exam. Yes, 1.5. How many can you produce with an NADH? 2.5. We have synthesized up to this point how many ATPs? Okay, pull out your calculators. How many have we produced? You get 1.5 from FADH. Then there's 4. That is wrong. Why do I say that is wrong? You're actually you've made 2. Because what? It cost me energy where? To break down this fat. It went from the fat broke down into fatty acid, got into albumin, went into the bloodstream, got to the cell, the muscle cell, was transported into the cytosol, and it was a free fat in the cytosol. Then it had to be transported into the mitochondria. What was the first step for us to cause the transport to occur? We had to add a CoA. How many phosphates were used to do that? 2 equivalents. And so I used 2 before I even got in, so I'm -2. At this moment in time, I have synthesized equivalents of 4, and so at this moment in time, I've only made 2. Okay? I now have this 3-keto acyl-CoA. All right. Talk to me about what you would think about the acidity of these protons. They're pretty acidic. We said that if it's alpha to a carbonyl, it's acidic, right? And it's alpha and alpha to a carbonyl. This is an anhydride, so they're really acidic. And so that's kind of that's an organic structure and what you get, but the enzyme doesn't take off any of those protons. In actuality, I'm going to pick up a proton there. What really happens is thialysis. Hydrolysis is a water that gets deprotonated and then attacks. Thialysis is a sulfur compound that gets deprotonated. It will attack right here. It will kick up, it will kick down, and this will pick up a proton. And what we have falling off is an acetyl-CoA and an acyl-CoA that's just 2 carbons short. Okay? That acetyl-CoA then can enter in the citric acid cycle and be oxidized completely to CO2, producing lots of NAD pluses. It can produce a GTP, which is an ATP equivalent, and it produces some FADH2. So there's a lot of energy potential right there. And I'm shortened by 2. That fat then can go through the 4 steps again. Oxidation, hydration, oxidation, thialysis. And then it can go again, and then it can go again, and then it can go again. If I had a normal 16-carbon fat, how many rounds of this cycle do I need to break it all into acetyl-CoAs? 16 carbons, breaks off 2, breaks off 2. You would think 8. It is actually 7. Because in the last step, you would have 4 carbons. In the 7th step, you had 4. When you do the thialysis, you'll end up with 2, the acetyl-CoA, and that will be an acetyl-CoA. So it only takes 7. So wherever you have the number of fat, the number of rounds it takes is N, 16, minus 2, 14 divided by 2. So N minus 2 divided by 2. I give you a whole slide for that. Okay, and in terms of stoichiometry. The first enzyme have different classes, long-chained, medium-chained, and short-chained. So that first oxidation can happen based upon which enzyme it binds to, and you can go through all of the degradation. The last 3 enzymes are much more generalized. It didn't matter how long the fat was, they can continue the reaction. But the acyl dehydrogenase is specific. This also leads to potential diseases. Okay? If you are absent one of the 3, which one do you not want to be absent? One is more fatal than the others. And there's one that is the most tolerant. Okay, anybody got it? And would you say, oh, so would it be long chain dehydrogenase, medium chain dehydrogenase, or short chain dehydrogenase? Okay, got a medium, somebody right in the middle. Long, got someone say is long. So one here, one there. Short, oh good, I was hoping. All right, 1, and 1. Oh, 2 on the mediums. A short. Okay, yes. If by chance you had a disease in which one of the dehydrogenases was inactive or not synthesized, which one would be most fatal to you? Okay, good. We're right smack dab in the middle. Everybody's guessing. All right. Okay. It is long. Can anybody tell me why? Say that again? Yes, it is more carbons not being broken down. But the reason why is that what is the most often seen fat? 16 carbons. And if you can never break down the 16, it will never become 14. And so even though you have medium and short chain enzymes that are functional, you'll never see those fats because you can't process the ones that are most often there. And so if you ever have to rely on your fat stores, you'll never, ever be able to extract energy from them. And so long chain, people who have issues with long chain are very rarely seen. There are people that have diseases that have impaired medium and short chain. And if you actually had to have a disease, you hope it's the short chain because then you can extract all the way from 16 down to 4 and extract most of the energy. You just have higher levels of that 4 carbon to intermediate that become an issue in terms of getting rid of it. It'll build up in concentration. So each round produces lots of electron carriers and an acetyl-CoA that can be broken down. The number of degradations requires N-2 divided by 2 reactions, and it yields N-2 divided by 2 FADs times 1.5 ATP, N-2 divided by 2 NADHs times 2.5. You get N-2 divided by 2 protons, and N divided by 2 acetyl-CoAs, and through the entire oxidative process in the citric acid cycle, you get the equivalent of electron carriers and GTP. The equivalency would be 10 ATPs. For each fat, you have to substitute 2 ATPs for its activation from where it was carried in the bloodstream. And so you can get lots of energy. All right, here's the deal. You can degrade the straight chain, but we also have unsaturated fats. And this double bond gets in the way. And so the double bond that a lot of times that we have naturally are in the cis conformation. And in the process of beta oxidation, we have to get to a trans. And so that's an issue in terms of the reactivity. And it's more complex. Okay? We have to get it into the trans 2 to be in line with the degradative process. We have 2 cases, whether the unsaturation is in an odd carbon or an even carbon, because we have to have cases or steps to handle both of those. So what happens is your fat will be oxidized, go through beta oxidation, until it gets close to the unsaturation. In the odd case, wherever it is, it will degrade until you get to a cis delta 3 in that position. And in the even case, you will degrade normally until you get the double bond in a cis delta 4. Each one of them have to be converted into a trans 2, and I have to use different enzymes to get it into that. When you have the cis delta 3, there is an isomerase which can convert this. And remember, isomerases is deprotonate and protonate until I get where I need to go. So I'm going to articulate this proton. And to do the transition, if I look at the starting material and the ending material, 2 here, 1 there, obviously I'm going to have to take 1 out. These electrons can form the double bond, okay? And I can pick up a proton, okay? The intermediate has a negative charge on that carbon. All right? That carbon sits out there, it's got a negative charge carbanion, really, really bad. Unless we said it was alpha to a carbonyl, right? So, that is really, really bad, yes or no? Can you see something that actually stabilizes that carbanion? It's not alpha to a carbonyl, right? But temporarily, there is a double bond in that mechanism here. And so what I could do is conjugate through that double bond, and that is an equivalent of being alpha to a carbonyl. So having that carbanion in that particular position is not as high in energy as you might predict, because it does have a resonant state. Now that resonant state really doesn't exist too often because, again, the proton that's going to react is going to be there very quickly, and I get into the trans 2. At this point, I'm done. I can go on with my reaction. All right? A fat that is in the odd case, though, reduces the amount of ATP that can be synthesized, because I have an unsaturation in which I didn't need to use the first dehydrogenase. So I will create 1 less FADH2 in the degradative process for that double bond, and so I won't extract the energy as much as if it was fully saturated. Okay? So I lose 1.5 ATP. Write that down somewhere. It's going to come back. All right? In the even case, it's a little bit different. I get to the place where the delta 4 unsaturation comes out. Now, it will react with the dehydrogenase. I will make an FADH2 in the even case, and I'll get a conjugated double bond system. So I extract as much energy as if it were saturated. The next is a reductase that uses the energy of NADPH, and when I see NADPH, I think mechanistically an H+, a hydride, and what I'm going to do is I'm going to attack this. The electrons will go into the 3rd position, and this will pick up a proton. The last arrow would have a negative charge alpha to a carbonyl in the mechanism, so that is stabilized. And I now have a 3 version of the unsaturation, which we just learned can be isomerized and enter in. In the case of an even unsaturation, I will extract as much energy as a fully saturated, but that is only done because I introduce energy from NADPH. So I have to take energy from NADPH, which doesn't create any ATP directly, because it isn't used to carry electrons in the synthesis of ATP, but it is an energy source usually used in synthesis, and so energy is transferred. What this would require me to do, the energy is extracted elsewhere. I would have to take one of my glucose molecules and send it down pentose phosphate pathway to make a ribose, and I would lose energy in terms of there's less glucose to extract energy from. So that's where the energy is lost. And that isomerization gets us into a normal intermediate. So you need to think about how an odd and even case works. Okay. Here is the wording of the mechanism that we looked at and in the slides. And so it just outlines the steps by which you bypass the unsaturation. All right. Every so often, you will find an odd-numbered fat. Not a lot, but we introduce it here. We introduce this concept here because there is fats that are odd, but it's very rare. We will see propanil-CoA, though, synthesized or as part of the degradative process of amino acids. And so here is where, in our next chapter, as we degrade amino acids, this is a common place where fats and amino acid degradation come together. Because we introduced fats first, we're going to show the very rare case, but in proteins, most of the degradation and most of the propanil-CoA comes from amino acid degradation. All right? And so odd fats actually come in the form of bovine milk that we drink. This is where the largest source of those come in. The propanil-CoA will be carboxylated into methylmalonyl-CoA. This is very similar to taking pyruvate and reacting with CO2 to make oxaloacetate in gluconeogenesis. Same mechanistic steps. The demethylmalonyl-CoA, however, is not real usable. We actually have to convert it into the L isomer for it to be processed. All right. Again, even the methylmalonyl-CoA is not very suitable, but we're going to transform it into succinyl-CoA. What pathway am I here? Citric acid cycle. So it's going to enter in the citric acid cycle. Now, look at that reaction. What has happened? The carbon of the acid is attached to, okay, if ever you're looking at things, you need to start inventorying, and I will start that. CoA, 1 carbon. CoA, 1 carbon, right? Then the 1 carbon over here is attached to a carbon. That must be the 2 carbon. This must be the 2 carbon. And then the 2 carbon over here is attached to that carbon. So this is 3. And it's not the carbon here that is attached to the oxygens. So the only one that is not attached is this one. And so this carbon that has the oxygen is 4, and that is 4. That doesn't look great. At times, though, in organic chemistry, you may have been introduced to reactions in which a hydride shifts or even a methyl shifts. Here, I'm not shifting a hydride or a methyl, I'm shifting an acid. That's kind of weird. Do you remember what kind of reactions tend to allow hydride shifts and or methyl shifts? Very special type of reaction. Oh, I'm going to have to get on Dr. Oden's to make sure. How many people took Dr. Oden's for organic? How many people took Dr. Brown? Nobody? So okay, so it's all up to Dr. Oden's here. Ooh. Oh, who took Hearn? God bless you. You got through. Okay. Our PChem is teaching organic. We're going to go through a radical mechanism, and that's where the shifts actually occur, through radical mechanisms. We need in this a vitamin B, and the vitamin B is vitamin B12 to do this conformation. Now, I do like you to know vitamin structures and maybe reproduce them. So this is one that I would want you to draw out on an exam. No. This particular vitamin B12 called cobalamin is rather complex. You do see a heme ring, right? Or the porphyrin ring that looks kind of like a heme, but in the middle we've got cobalt. In photosynthesis they had magnesium, and in our bodies we have iron to bind to oxygen, and we got all sorts of things happening. The cobalt is coordinated to 6 different species. 4 of the nitrogens in the ring, the nitrogen of this item at the bottom, that cofactor at the bottom, and then at the top we got a coordinating event. All right, one of these bonds, sesame seed, one of these bonds doesn't look like the other. One of these bonds is just not the same. Which bond is not the same? And if you took GChem 2 with Krebs, he loves metal centers, and he loves the coordination, and he's an inorganic chemist, and he barrels down GChem 2 so he can get to that. Metal centers love to attract what? What was that? Electrons, yeah. Lone pairs. And so a nitrogen has lone pairs, right? And 5 of these bonds are being coordinated through a lone pair through nitrogen. One of these bonds doesn't normally have a lone pair sitting on it. Which one is that one? The cobalt to the what atom? Do you see it? What do you see? It's a methyl group. Methyl groups don't like to have a lone pair to donate to something. Okay, so the bond between the carbon of the methylene group and the cobalt is a very strange bond, not necessarily normally seen coordinated to metal centers. This is where the reactivity is. Okay? It's actually a large cofactor, and it is not absorbed as easily as you might think. The reaction takes ATP and creates that carbon bond. And so if I'm reacting with ATP, do I attack at the alpha, beta, or gamma phosphate? None of them. That's a trick question. Because the leaving group is all 3 phosphates. It has to react with the carbon. A triphosphate is a leaving group. This is really, really strange. But it is also a really, really strange bond in which a carbon is coordinated to a metal center. It's a weak bond. Now, when we break bonds, almost always we write an arrow with 2 heads called a homolytic cleavage, where they go together. But you can also have a breakage in terms of a heterolytic cleavage, in which 1 electron goes, and it's a half arrow, and 1 electron goes, and it's a half arrow. And this really strange bond breaks weirdly, and it leaves a radical on that particular methylene. And so that entire huge group, okay, reacts in such a way to provide a carbon radical. And did you see that whole slide that took up that whole thing? Well, now we just call it R. Okay? And we have a radical. The radical will react with this electron pair. We will show the reaction this way, that I will take this one and put it in towards a bond, and I will take one electron and put it towards a bond, and one of the electrons is left on the carbon. And I have a radical now on the substrate. Okay. The next reaction, okay, is what is going on? All right. I said the acid shifted. Well, it sure looked like it, but it really wasn't the acid that shifted. It was actually the carbonyl. And so I'm reacting this radical with this bonding pair right here. All right. So I will do it this way. I'll put one electron to make a new bond. I will take one electron to make a new bond. And I will take one electron to be on that carbon. So this bond right here are the two radicals reacting together. I now have them all in a straight linear chain. The last step in a radical mechanism is a terminating step. And I will take one electron from the cofactor, that's the vitamin, and one electron to create the bond that is seen here. And I've terminated the substrate. I now have a reactive catalytic cofactor. And I have succinyl-CoA. All right. This cofactor is huge. And if you actually take a look, we're also reacting it with CoA. CoA is huge. The substrate in this entire thing is that little tiny thing right there. And do you see the rest? That's all cofactor. And this is the CoA. And it takes up a very large volume to carry that little tiny reaction. Okay. All powerful in an itty-bitty space. Now, the lack of this vitamin leads to a disease, okay, called pernicious anemia. And it's because this very complex molecule is only synthesized in plants by microorganisms, and we have to ingest it. So you eat this thing. Once it gets into the gut, there's an intrinsic factor that has to bind to it to get it into the bloodstream. It's a very complex internalization. Even when it's in the bloodstream, it has to be carried and it has to be delivered inside the cells, and in this case, the reactions occur in the liver, by a receptor-mediated event for endocytosis to get it inside. Okay? The absence of cobalamin in the liver cells is what causes the disease. The only treatment that we have is to inject cobalamin at monthly intervals directly into the muscle. Okay? Most of the time, it's the lack of the IF factor. We only need about 10 micrograms of cobalamin every day, and so people respond really, really well if they have the right disease. There are some that have cobalamin injected, but it doesn't help them. The factor for them is that there is an error, not in the fact that it doesn't have the intrinsic factor, but the cobalamin 2 transporter is inactive. And so some people respond to injections and some people don't respond to injections. Okay. If you cannot get rid of or react methylmalonyl-CoA, you tend to get acidosis in the bloodstream because these are acids that are build up in the bloodstream and create a more acidic environment. This also, all of the blood chemistry, enzymatic reactions are off because the pH of the blood is off, and you can detect it by having this particular molecule being excreted in the urine. You should not see any of this in the urine at all.